Okay, well, let me make a little introduction. I am Juan Ospina, bacteriologist, PhD in biotechnology. I have been interested in Aptámeros for about 8 years now, specifically for the diagnosis of the tropical diseases. I'm currently writing a book about Aptámeros, and that is why we are here today with Dr. Szostak, who is one of the pioneers of this technology. Okay, Dr. Szostak is a professor of chemistry and chemical biology at Harvard University, professor of genetics at Harvard Medical School, an investigator of the Howard Hughes Medical Institute, and the Alex Reese Distinguished Investigator at Massachusetts General Hospital. Throughout his career, Dr. Szostak has focused on nucleic acid biochemistry, including seminal work on DNA repair, telomerase and telomerase, and the laboratory evolution of aptamers and ribosomes. He is recipient of the 1994 National Academy of Sciences Award in Molecular Biology, the 2006 Albert Lasker Basic Medical Research Award, the 2008 H.P. Heineken Prize in Biophysics and Biochemistry, and obviously the 2009 Nobel Prize in Physiology or Medicine. He's a member of the National Academy of Sciences, the American Philosophical Society, and was recently elected a Fellow of the Royal Society. Dr. Szostak current academic research is focused on the origin of life. So I want to take this space to clarify 6 questions that I would like to ask you. I had the fortune to talk to Dr. Larry Gold a little less than a week ago. He told me about his experience in what he called the twerk moment, as it was his postdoc student who performed the experiment that will lead to the discovery of the selection strategy. We know of your great work between 1980 and 1989 with Jess that will lead you to win the Nobel Prize. And from then on, your interest in RNA began. So the first question is, could you tell us about your work with RNA with Andy Ellington? How was the work carried out? And how did you take the conclusion of this novel discovery?
Okay, sure. So we started working on RNA biochemistry, mostly ribozymes, around 1985. And my first student to get involved in this was Jennifer Doudna, who's now famous for her work on CRISPR. With Jennifer and several other amazing students like Rachel Green, we tried to make use of naturally occurring ribozymes to do replication-like reactions, sort of following up on the RNA world idea that life might have begun with self-replicating RNAs. And that was it was very interesting and promising, but eventually we came to the conclusion that we really needed to be able to evolve our own RNAs, because the ribozymes that are found in nature are not really, meant for, they're not evolved to do replication chemistry. So that led to the idea of trying to find new RNAs that could act as catalysts in pools of random sequences. And we thought we should start with something simpler, which is just trying to find RNA sequences that would bind to some molecular target. And what made me really interested in doing that was that when I talked to some people, typically people who worked on, say, protein structures, they would say that the chance of finding a functional RNA, something that could fold up and make a binding site just in random sequences would be almost impossible, like probability of 10 to the minus 50th. Of course, we can only in the lab look through, say, 10 to the 15 or 10 to the 16 sequences. But other people that I talked to would say, oh no, it's going to be extremely easy. They would say that the solutions, sequences that could bind a target might be, very common, maybe 1 in 10 to the 5th. So that seems like a huge range of uncertainty, right? Between 1 in 10 to the 5th and 1 in 10 to the 50th. It's a big range. So, talking with Andy Ellington and other people in the lab, we thought it would be worth exploring that. And so Andy Ellington set out to actually do the experiment, and we realized that, although we could make large collections of random sequences fairly easily, we wouldn't be able to purify an incredibly rare molecule out of those pools all in one purification step. So we thought it would make more sense to do what's now known as SELEX, but basically enrich for molecules that do the binding you're interested in, then amplify those surviving molecules, then enriched again, and go round and around this cycle. So yeah, it worked. It was not easy, but Andy developed the method in my lab, and he was successful in finding RNAs that could bind to small molecule targets, and so that led to a huge explosion of work after that, and yeah, eventually finding catalysts and other more interesting molecules.
Yeah, right. The second question is, how is possible that we can manipulate the aptámeros to recognize a specific target if the nucleic acids have a pre-established function for years of evolution?
Sorry, I didn't quite understand the question. Could you say that again?
Okay, how is possible that we can manipulate the aptámeros to recognize specific targets if nucleic acids have a function established for years of evolution? How is possible that we can train these RNAs to recognize something? What is the explanation of that?
Well, I think it's, in a sense, it's nothing really special. When you have a sequence, and it's a sequence of nucleotides, if the sequence is the right sequence, it will spontaneously fold up into a three-dimensional shape. It will project its functional groups in the right way to interact with the target molecule. And I think the right way to look at it is, it's not like we're training the RNA to do this. We're finding the sequences that exist, okay, that can do any tasks that we set them to, such as binding a target, or whether that's a small molecule target or like a drug, or binding a protein. Those sequences exist in sequence space. It's just a matter of finding them. And when you find one, of course, you can introduce mutations and try to optimize it so that it binds more tightly or catalyzes the reaction more rapidly. But it's like a process that optimization is Darwinian evolution, right? It's just narrowing down and finding the best sequence that can do a job.
Okay. In the third question is, in vitro evolution is one of my favorite terms to describe the SELEX strategy. Who came up with the idea of using this term, in vitro evolution and aptámeros, and why?
So the term aptámeros, I think, actually came from a friend of Andy Ellington's. and it comes from, so some people don't like the term because I think the first part, the apta, or comes from Latin, aptus, which means something like to fit. And then mer is, a Greek suffix. And so it's a kind of hybrid word. But I think it's a great term. It's a descriptive and easy to say, and its origins means basically an oligomer that fits the target. And the idea for in vitro selection, as a way of sort of finding very rare sequences and then continuing from in vitro selection to in vitro evolution, it's kind of the same process, except that you introduce more mutations and continue to select for better activity. And I think, we were all, our background, my background especially, was in genetics. And so, I think we've been basically doing evolution either, with organisms, or, I worked on yeast, and so we did many selections on living organisms. And the idea of doing a selection on molecules, it's not really that different.
Okay, the question 4 is, throughout the years, what has been the practical application of Aptámeros that most caught your attention?
Yeah, so I was always more interested in the fundamental implications of aptamers and the ability to select for aptamers and ribozymes. So, I kind of recognize the idea that you could use aptamers like antibodies. They're essentially an RNA version of an antibody, right? So in principle, anything that you could do with antibodies or, various protein binding domains, in principle, you should be able to do with RNA aptamers or DNA aptamers. But it's taken a lot of time to develop that, and to develop the chemistry, and to solve a lot of the problems. If you want to use them as a therapeutic, it's there's very, very, very limited examples. I think the easier practical application are in various diagnostic approaches. And I think they have a lot of advantages there, because you can chemically synthesize aptamers. You can make very stable, chemically stable versions of them. So I think for the time being, the diagnostic applications, I think, are important, and I hope they continue to be developed. I think a lot of people are developing so-called apta sensors as a way, so using an aptamer as a recognition domain, perhaps coupled to other domains that give a signal, a readout of some sort. And so the idea of getting real-time readouts of biological signals, I think, is very interesting.
It's amazing. Okay, the 5th question is, after evaluating the potential of aptamers, you focus on, you work on discovering aptamers of a protein nature. You were never interested in commercializing this technology, or were you only interested in knowing the basic of the origin of life?
Well, I think I was always more interested in understanding the implications for the origin of life. For RNA and DNA aptamers, I didn't pursue commercial applications of that, because I thought it was going to be very, very difficult and would take a very, very long time to work out all the many problems, which I think is true. they are being gradually worked out, and I think obviously Larry Gold's company has made a huge amount of progress. So they're doing some great stuff with large collections of aptamers. But I did, with Rich Roberts, also develop a way of selecting for peptide and protein sequences. So some people call those peptide aptamers, or it's just another application of in vitro selection. And so for that, in that case, we did pursue commercial applications and actually started 2 different companies to sort of develop those applications. So that also took a very long time. One company basically didn't work out. The second company was more successful, but was also bought out recently. But I think those companies have, they did some good in developing useful molecules.
Nice. Okay, the 6th question is, obviously, I want to ask you about the RNA war hypothesis. We have more and more evidence of its feasibility. What, according to you, will be the definitive test, the artificial cells that you are creating?
Yeah, so we're trying to develop or to create or build in the lab very, very simple cells that we think might look something like the first cells, primordial cells or protocells. So they basically would consist of a self-replicating collection of RNAs in a membrane envelope, and where the membrane envelope also can grow and divide. So I think we're fairly close to being able to build something like that. The hard part has been demonstrating RNA replication without the use of any RNA polymerases or ribozymes, right? Because at the time of the origin of life, there were no proteins, no enzymes, no ribozymes. All of that had to emerge spontaneously. So yeah, so what we're hoping is that if we can actually have a chemical system that will allow for RNA replication in replicating protocells, we should see, and we would like to see Darwinian evolution emerge spontaneously. So that's the goal. It may still take a while, but I think we're making progress.
Amazing. Your work is amazing. I have more additional question, but it's so simple. What did you feel when you received the Nobel Prize?
Yeah, well, it was, it's obviously is a pretty exciting moment. And so, yeah, I was very, very happy about it. It wasn't maybe a total surprise, because people had been talking about it as a possibility for a few years. But obviously it's very wonderful to have that kind of validation or recognition of one's work. So yeah, so that was very nice. But yeah, since then I've concentrated more on questions like in vitro evolution and the origin of life, because I think these fundamental questions are the most interesting things to me.
Of course. Dr. Szostak, thank you very much for agreeing to this interview. It's a real honor for me to meet you.
It was very nice to meet you. Nice talking with you. Thank you.
As soon as my book is published, I will send you a copy.
Great. Thank you very much.
Infinite thanks. Bye-bye.
Okay. Have a good day. Bye.